just doing the chapter announcement so we can get to what you've been waiting for. This is a very exciting night, so welcome everybody. Let me just get these slideshows started, and we can begin. All right, so welcome everyone. Once again, my name is Jared Nigro, and welcome to the May monthly meeting for Pasadena Audubon Society. What you're looking at is an island scrub jay, thinking, I've got it pretty good. And I'd say so. It's a beautiful, healthy bird. I think it's had a pretty good life. Scorpion Canyon Campground from February 10th, 2025. And this is from Dominic. And this beautiful, beautiful photograph. So welcome everybody. We've been very busy this past month since you've last seen us in April, and so I'm gonna jump in and tell you what we've been up to and what's ahead. So first things first, May has been an excellent month, thanks to our new field trips chair, Alex Coffey, and the whole team behind all of the bird sits and all the field trips. Thanks everybody who goes on them, who leads them, who coordinates them. Thanks to Alex for taking this over. It's very generous. This is a big job, and he took it on. And I just met his 3.5-year-old today. So he's raising a 3.5-year-old and planning these field trips. I mean, give that man a trophy. So we have a lot of great field trips coming up for the second half of this month. Last weekend was Santa Cruz Island. That was a project trip. It was the bird count. If you haven't seen, the bird count was great. The species count in general was excellent. It was a huge success. And if you're interested in seeing that and you haven't seen that yet, you can just email us and we'll forward along that information. But I heard it was very, very birdie, a very successful trip. So to see the second half of the month's, sort of the second half, last third of the month, just go to pasadenaaudubon.org/fieldtrips and sign up for some. June, July, August, it starts to get extremely warm. It's less birdie. Everyone's less interested in being outside after like 9 a.m. So the field trips will be winding down for the summer months, as is always the case. So try to catch these last few in May if you can. So this was a very exciting, I'm sure you all saw this was a very exciting flash fundraiser. Bins to Ghana. Boy, we did not plan this. This just came in, and you all, I can't thank you enough. That we raised enough money to supply, I believe it was 10 binoculars, brand new binoculars to Ghana in less than 3 hours. Folks were so generous. This is going to, this came to us from Graoping Hong, who is a professor at USC, and he told us about this opportunity to assist this to steward birding in this area, in this lagoon in Ghana. And they have quite a bit of education programs popping up. The local university is expanding their birding. And so they all formed this sort of coalition called the Lagoon Committee. And we are now supplying them with binoculars. And this is the third year in a row that we've sent our bins to somewhere outside of the US. So we're very proud of that. This was definitely our shortest campaign, but incredibly successful. So I can't thank you all enough for your generosity in supporting this in such a quick amount of time. And speaking of generosity, it's hot out and people are still weeding. This is so kind. Hahamanga Habitat Project. So if we're going to do another work day coming up, and I'm going to spoil some, we're working on very cool Hahamanga t-shirts. So if that's not a reason enough to show up and pull some invasive plants, I don't know what is. So this is at the Watershed Park in Pasadena. They are going to announce the next work day, but it does start at 8 a.m. So you don't, boil over in the heat. And we just need, as much help as we can. You can bring your own tools. You can share with others. No experience needed. You can always contact Darren Dow, our former board member and field trips chair. He's still dedicated to the Hahamanga project. And so you can always email him to sign up and learn more. Oh, Birds and Beers are our continuous party, our ongoing party. So this is this is the cure to having Zoom meetings. This is Birds and Beers. This is an in-person social event. Next Wednesday, the 28th from 5 to 8 p.m. It's at the Wild Parrot Brewing Company in East Colorado Boulevard. There are no themes. Carl has done a great job of having a theme every year. Or theme every month, sorry. We had some great jokes in honor of Dave Weesoff and all his bird jokes. But this time we're just having fun. So please come support. It's a great way to meet fellow birders. And the most important thing is they donate $1 for every pint sold. So we get a little bit of cash, and that just helps us run. So thanks everybody who comes to that. And please, if you ever considered it, if you're ever nervous, like, oh, I don't like socializing, we're a very friendly group. So please come. And I think this is my second to the last announcement. Okay, so this is the June banquet, which I guess these tickets are selling out fast, which is awesome because last year we were kind of hoping around this point, we were hoping that we would fill the house. And this year it looks like we are filling the house. Now, obviously, we all know that it's not going to be at the Eden Canyon Nature Center this year. The Neighborhood Unitarian Universalist Church is the new venue that's right next to the Gamble House. You can see our Hummingbird Cantina before you come over. So whether or not you plan to attend, we would still love you to have we'd still love you to submit your bird photographs so we can use it for the bird trivia slideshow. All you have to do is just email us 3 of your favorite bird photos with a brief description of each. Of course, include your name so we can credit you. PasadenaAudubon@gmail.com. In the subject line, just bird photos for banquet slideshows. Please, you might say, I'm not a great photographer. That's not true. I'm sure you're great. Send them in. We're happy to have them. And once again, this is Saturday, June 28th, 6:30 to 8:15 p.m. Please come if you've never been before. We really have a great time. We have a lot of food. I just put in the order for the food. So we're going to have great Mediterranean food and baklava and cake. So it's going to be a great time. And now I'll turn it over to Chris to talk about the June monthly meeting.
Yes, indeed. Next month, we're again a Zoom-only meeting next month. And next month's speaker is John Garrett, who actually some of you may know. He grew up here in the Pasadena area and was a very active birder here for the time he was here. But now he's at Cornell University, where he is a big muckety muck in eBird. And so he is going to join us to first of all, to answer any and all of your questions about eBird, how it works, how you can use it more efficiently, how you can do more with it, and he'll also give us kind of a behind the scenes look at how this immense project, arguably the biggest citizen science project on the planet, manages all those checklists that folks like you were sending in. So that's Wednesday, June 18th, online at 7 o'clock. Hope we see you a month from now. But for now, let's move on to tonight's topic, and this is a topic that I've wanted us to present for a long time. I think it's interesting and it's somewhat controversial, and I hope everyone enjoys it. As I was planning this talk, the thing that kept popping up in my head was that for the past, I don't know, half century of the modern environmental movement, there have been so many successful conservation campaigns that have relied on a simple 3-word message. And those 3 words being extinction is forever. It's a very powerful statement. But what if extinction wasn't forever? What if we could actually bring supposedly forever gone plants and animal species back? Well, that is the topic of tonight's speaker. And our speaker tonight is at the forefront of these attempts that kind of mix science fiction and actual science fact. Tonight's speaker is Ben Novak. He's the lead scientist for the conservation nonprofit group called Revive and Restore. He manages nearly 24 projects developing biobanking, reproductive, and genome editing technologies for birds, of course, but also for amphibians and mammals, including, as he's going to talk about tonight, the audacious effort. Audacious is not too strong a word. effort to recreate the extinct passenger pigeon, and he says we might be able to do it by the early 2030s. Uh, a little bit about Ben. He has a bachelor's degree in ecology and evolution from Montana State University, a master of arts in ecology and evolutionary biology from the University of California at Santa Cruz, and he leads the world's first genetic rescue cloning program that has already produced the first clone of an endangered species of black-footed ferret. But tonight he's going to talk about going one step further, not reproducing something endangered, but reproducing an honest to God extinct animal, the passenger pigeon. Quick reminder for everyone: if you have questions for Ben as the night goes on, please leave them in the chat. And with that, I'd like to turn it over to Ben Novak. Welcome.
Hi, thanks for having me. Well, I'll spoil a little bit first, because I hate to spoil what Chris said, but the new passenger pigeons won't really probably be honest to God passenger pigeons, but we'll show why in a minute here. So I'll share my presentation and get going here. So this is about the new passenger pigeon, and we'll talk a bit about the science. Now, I don't have any cool actual data to show you, partly because I'm not the scientist doing the work in the lab. Those are a team of other people. And right now it's all genomes and stuff, so it doesn't really look like much to look at anyway. But I'll start by talking about Revive and Restore, who I work for. I've been working for Revive and Restore since it's founded in 2012, and it formed around this concept of biotechnology for conservation. The co-founders Ryan Phelan, Stuart Brand. Stuart Brand co-founded the Long Now Foundation. Was part of the early environmentalist movement, but has always been a techno-optimist. And they saw what biotechnology was doing in medicine and other things, and thought, they better bring this to conservation. So, conservation, as I think everyone knows it, is about protecting habitats, protecting species, restoring ecosystems. There's a lot of tools to do that. And as Chris said, there's been a lot of success stories that get overshadowed a great deal by the fact that we are technically still, not technically, we are losing the war for biodiversity right now against those that exploit natural resources, which is basically everything in the world as we know it. Um, and there are these problems that are just getting worse. Invasive species are getting worse. Disease spread by those species, as well as emerging disease from climate change, is getting worse. And now, after centuries of biological annihilation, we not only lost a lot of species, but we've lost genetic diversity in the species that are still surviving. And all these challenges are being exacerbated by climate change, and climate change is bringing its own direct impacts as well. So these are really challenging problems, and it's really difficult for those conventional conservation tools to succeed. If you start by just trying to protect habitats, moving animals around, etc., sometimes those approaches don't really work very well, and there can develop inertia. The status quo can at times be slow to want to adopt and try new things, and instead they keep trying the same things they've always done, despite the fact that they're not working. The success gets only so far, and when you've tried something over and over and over again and it's not working, that's when your donors start to fall out. Even I've even seen heard of stories from Fish and Wildlife Service where basically because an intervention didn't work after multiple tries, they weren't allowed to do it anymore. So these barriers come up. And the worst case scenario is that when you start out to solve a problem and save a species, you end up at extinction anyway. Just because these threats are so intractable and difficult to overcome, especially at scale. So our theory of change at Revive and Restore is to create what we call the Genetic Rescue Toolkit that is built on research and development and innovation. It is all about getting to those new tools and adding those into the conservation mix. Hopefully, these things can inspire people rather than make them feel deflated, and make people want to do more and lead to the goal of saving and restoring ecosystems. The genetic rescue toolkit is basically a whole bunch of tools that rest upon genome sequencing and just the DNA data that we get from species. That does a lot. The majority of our projects we fund around the world have simply been in genomics. But when you start adding layers onto it, such as cell culture, in vitro technologies, biobanking, and then advanced reproductive technologies, you can do things like clone a black-footed ferret from 1988 to bring new diversity into the population. You can then add things like genetic engineering and gene editing once you have all those other tiers, and then you can start talking about things like de-extinction, but a lot more. Combining these tools together is to try towards that ecological goal of restoration. How do we do that? Well, if you mix and match these tools in the right ways, you can accomplish several things. One is restoring diversity, particularly that genetic diversity, in ways that no other tool can. And then doing things that no conventional tool has ever been able to do, which is facilitate adaptation by giving species either new genes or helping spread the genes they have that confer resilience to disease or climate change, and then bringing in humane and scalable ways to control invasive species. These are the big buckets that these new tools accelerate and open up new opportunities for. We are a nonprofit, and what we do is we try to identify the white space. Where are the opportunities? What isn't being done or applied? We fund groups around the world as well as design our own projects to advance the technologies. And then we try to take things that have been established, such as just genome sequencing, which has been around for a long time, or biobanking, and get those actually out there and being applied. So it's all about new science and getting existing science rolling out. We have 80 projects to date. 30 of those have just completed in the last few years. These are funded through institutions in 21 nations that are helping 199 species around the world, either by just adding new genome resources or by fundamentally developing new cell culture techniques and other technologies. I am responsible for what could be considered four programs: our conservation cloning work, our informed biobanking work with Fish and Wildlife Service, um, the de-extinction work, and the biotech for bird conservation program, which is aiming at developing a suite of reproductive and gene editing technologies for all birds, not just for bringing back the extinct passenger pigeon, which is what I'm probably most famous for. So, the passenger pigeon. You guys are out in California, but I imagine you probably know a lot about it already anyway. But I always like to talk about what this bird is. Um, that is a real photograph. It's one of the few extinct species that lived long enough into modern history to be photographed. That's a male, really nice one from the side. I really like this quote from Chief Pokagon of the Potawatomi tribe. He said, "It was proverbial with our fathers that if the Great Spirit in his wisdom could have created a more elegant bird in plumage, form, and movement, he never did." That's certainly what I felt in my heart the first time I saw a photograph of a specimen and when I was 13 years old and I fell in love with this species. And then I read about its story, and what captivated me was basically summed up by this quote by John James Audubon. When he witnessed a flock in 1831, he said, "The air was literally filled with pigeons. The light of the noonday sun was obscured as by an eclipse." Because that's what this bird was. Unlike most other birds, it was hypersocial, never disbanded its flocks really. And its flocks could be anywhere from several hundred million to 3 billion birds in size. The population was anywhere from 3 to 5 billion in the early 1800s at any given time. So there were only maybe two or three, maybe five flocks of these birds ever. So, despite being the most abundant bird in North America, it's estimated that in that time, one of every four individual birds on the entire continent was a passenger pigeon. So, of the 600-ish species that we know of that reside, at some point year round, seasonally or year round in North America, one out of every four was a passenger pigeon living in the eastern United States forests. Those flocks were really, really incredible. They would take days to fly overhead. And these are historic records that we have. We've accumulated every historic record we could get our hands on from the collections of other people and other things. And it gives you a nice solid idea that they were really an eastern forest bird. And their demise was swift. That's the second thing that made them famous. This species went extinct in the matter of decades through industrial scale trapping and shooting to feed the growing human population of the 1800s in the eastern United States. In 1914, the last individual, Martha, who lived at the Cincinnati Zoo, died, and it marked their extinction. It's also one of the few species where we know almost the hour that it went extinct, which was around 1 p.m., September 1st, 1914. So that's what made the bird famous and why people understand the story. So why do we want to bring back what was once the most abundant bird in the world? Well, it's all about the forest ecology. So in the eastern US, and actually most places of the world, forests are disturbance regimes. Um, you have a disturbance happen, something like a tornado or a fire that destroys some of the canopy, clears out the undergrowth, and then you get the successional regeneration of that patch. So this is showing one single patch of forest through time. And this is actually a figure from like the 1950s. This is really old forestry school science. I just gave it color and made it look pretty. Um, and the little bars there show types of species and when they occupy a particular stage in the regenerating habitat. So you can see that after a disturbance, a single patch of forest can host more species than a closed canopy would long term. Some species, like the eastern cottontail, which is endangered today, rely on those first successional thick underbrush habitats for their entire life cycle, while species like bats will like to roost in a closed canopy, but then will feed in those early successional habitats because they have more flowers and more insects, more things to eat. For a long time, it was believed that fire suppression in the early 1900s is what has led to our current state, in which those early successional habitats are extremely rare. They almost never happen without human involvement. So yeah, every once in a while there will be a severe storm, blizzard, or tornado that will kick this off. But at scale, humans are now the agent going out and doing this, and the scaling is not very good. All those species that rely on those early successional habitats are in decline today, despite the fact that we have more forest in the eastern United States than we've had for 150 years on average in anywhere. In places like New England, the forest cover has actually returned to what it was in the 1700s and the 1600s in some places. And that's because agriculture and mining shifted to the Great Plains and west. So those forests that had been chopped down, those lands weren't needed for human use anymore, and reforestation has occurred since the 1920s. But despite that, the species are still in decline. And as I said, for a long time, foresters thought this was because of the suppression of fire. However, our science that we've been doing for a decade now or so really points to more to the idea that passenger pigeons and their giant flocks were the long-term sustaining stimulus of disturbances. And it was their extinction that led to what we have today, which is lots of forest, but declining animal and plant species in terms of diversity. And that's because a giant flock of passenger pigeons would come into an area of forest and overcrowd the branches, break branches down, entire trees down sometimes. And in a matter of just a few days, if they're roosting, they would accumulate inches to possibly a foot of guano deep. And that guano would snuff out all the life on the forest floor. So, like a fire, it's clearing out the undergrowth. Like a storm, it's damaging the canopy. But unlike a storm or a fire, this is a tornado that's dumping poop. That's fertilizer. That's going to kickstart regeneration and that cycle. Now, this was never actively observed by people in the 1800s because they were really concerned with just killing the birds. But anecdotally, people would talk about how the vegetation was different in places where the pigeons had roosted. They would talk about how old pigeon roosts were the best and most fertile farmland to go after. So, from what we know from storms, from other birds today, this is our kind of theory of why we want them back, is that if we get this ecosystem engineer back, it's going to help with this forest cycle. So in the eastern United States, we should think of the forest cycle more so as in like the pigeon forest cycle. They come into an area, cause a lot of damage, and when they leave, the cycle starts. And because they were these huge flocks of billions, they really couldn't be strict migratory birds because there is no source of food that's going to feed a billion birds every single year in one area. So every few weeks, they are moving to somewhere new, wherever berries, seeds, whatever are plentiful. And therefore, their nomadic movements were creating a mosaic patchwork of these habitat stages throughout the forest, making a nice heterogeneous forest, much more diverse than it would be otherwise. When the pigeons were in a particular spot, dumping all of that poop on the ground, that was a huge resource for excrement consumers and vertebrates, which would be eaten by their predators at various levels. And basically, you could imagine there was probably entire behaviors and movements of species that are still alive today, but those behaviors and movements are gone now because of they don't have the passenger pigeon anymore. Um, it's possible that some predatory birds, like peregrine falcons, may have actually just followed giant flocks of passenger pigeons around because it was the biggest source of food. Um, and this would be basically an ecological hotspot wherever they went. And then when they were gone, that's when you would get that early successional regeneration where you're getting a lot of flowers, more pollinators coming in, and then that ecosystem shifting. If we bring these pigeons back, the idea is we make them abundant again, they start stimulating the forest cycle, and now we've secured the biodiversity of eastern US forests. That's the concept. But what is de-extinction? So I was asked to give this talk quite a while ago before de-extinction got in the news really, really big again in the last few weeks. I would assume I'll get a question about the news stories from the last few weeks when we do Q&A, but I really want to talk about the what and why for de-extinction, at least how it is practiced by us at Revive and Restore. So, when you think extinction, a lot of times you will think about dinosaurs. At least this is the slide I use when I talk to school students. But I think a lot of adults still think of dinosaurs with extinction. There have been five mass extinctions in history. The one that killed the dinosaurs is the most famous, but it was not the most severe. These are events in geological time in which more than 50% of all biodiversity on Earth gets wiped out in a matter of probably anywhere from a few tens to just 100 or so thousand years. It's a short time span, and the extinction rate goes much higher than background rates. And these have incredible effects on the evolution of life over time. Today, humans are causing what we as ecologists call the sixth mass extinction, and it started as soon as humans left Africa 130,000 years ago. Every single landmass they encountered, eventually, shortly after, you see the disappearance of all of the megafauna. Not all of it, but all of the super megafauna, most of the megafauna, the species that are bigger than human beings, basically. Species with big bodies that have a very particular role in nature. Now, what's interesting is when you look at mammals and birds, which we have the most data, only 5% of each group have actually been driven extinct by human beings over the last 130,000 years. But today, 25% are endangered and 12% are endangered. So we're not hitting that 50% mass extinction mark, right? But the rate is at mass extinction rates. And when we look at how many animals are in decline, in each group, over 50% of the species, whether they're considered endangered or not today, are in decline. And with birds, it's even worse because of climate change right now. So we're looking at potentially mass extinction event passing us by very soon. But the reason we've already technically hit mass extinction dynamics, and this is something you're not going to hear about a lot from a lot of speakers or a lot of articles, is because those 5% we've killed off have created an enormous eco-evolutionary deficit. Because, as I said, a majority of those species are the giant species, or the species that create huge ecological effect. It's a lot of species that were the only members of entire genuses, families, or orders. Some of the most unique and important species are the ones that we've killed off. It's been estimated in recent papers that the 5% of species we've lost to date would take anywhere from 3 to 7 million years of natural evolution to recover. And that 3 to 7 million years of recovery, that's mass extinction recovery times. That's what we saw after the dinosaurs, after the Permian extinction. It took anywhere from several million to 10 million, sometimes 20 million years to get back what was lost. We've lost 35% of our megafauna, 99% of our super megafauna in that 5% alone. That's what we're living through. And when people finally started recognizing that, we started developing conservation, and the passenger pigeon played a huge role in that. But 100 years ago, that's when hunting regulations and wildlife protections finally started taking shape. Nature reserves. 50 years ago, we passed the Endangered Species Act, and I hope every day we still have the Endangered Species Act because the Endangered Species Act is vital to restoring species, which is what people have been doing for a long time. So throughout the United States, this is a weird little diagram showing that where places elk have been reintroduced, or bighorn sheep, or wolves, or turkey. These are just a few examples. Species have been driven extinct locally, and thanks to conservation work, and even before the Endangered Species Act, but also made possible through the Endangered Species Act, people have been able to take species and move them from where they are still common and restore them to where they've been locally driven extinct. Sometimes only 20 years or so after they've disappeared, sometimes 200 years after they've disappeared. This type of reintroduction and rewilding work is something that's been happening around the globe. In a national park in Argentina, they've been reintroducing jaguars, giant anteaters, birds, tapirs. In Africa, they've reintroduced elephants and lions after they've been gone from an area for 100 years. In Great Britain, they've been reintroducing beaver, where they haven't lived in 400 years on that entire island. I got to see Oostvaardersplassen personally. It's an amazing place in the Netherlands where they've been reproducing the ecosystem that hasn't been there for 1,400 years. Tasmanian devils put back on the mainland to save them after 3,000 years. We have Pleistocene Park, an ecosystem being brought together that hasn't existed in 7,000 years. And the champion is the reintroduction of European bison in Spain, a place in which they have not lived in 15,000 years. Because time is not the number we care about here. It's not the component we care about. What we care about is what is the state of the ecosystem today? What are the roles that can have a beneficial impact to the future of life in that ecosystem? And the reality is that every species alive today, almost every species alive today existed 10,000 years ago. They existed 100,000 years ago. Most species existed a million years ago. The average lifespan of a species is anywhere from, say, 1 to 3 million years, which means all those species driven extinct over the last 130,000 years were somewhere in their 3 million year lifespan. They might have had a million plus years left to live. And all the species we have alive today have a longer evolutionary history living with extinct species than they've had time to adjust to living without them. And that's why you can literally just plug and play in a lot of environments, as long as you haven't had so much drastic change due to non-native species and other things that it's damaged beyond repair. We do this for a lot of reasons. There's the strict conservation reasons of increasing biodiversity, but there's also improving the environment, things like nutrient transport, pollutant filtering. And there's also basically the economy. Restoring species creates jobs. It also creates recreational hunting opportunities. It creates research opportunities and ecotourism. When people think about reintroducing species and restoring them, usually it's like in Yellowstone Park, where you have wolves that were driven extinct, but wolves still exist somewhere else. So you go and you get wolves, you put them back in Yellowstone. It's not the absolute original Yellowstone wolf, but but it's still a wolf. Then you have cases where a species is extinct, like the Mauritian giant tortoise. And you can't just go and find more Mauritian giant tortoises. They were endemic to Mauritius, but you can go to islands that are about 500 some miles away and get a giant tortoise that looks and very similar, eats the same things, and fill that void in the environment. This is called ecological replacement. And it's been done with a lot of tortoises on a lot of islands. And even though those species look extremely similar, they are diverged by millions of years in evolutionary time. They lived on islands that didn't even exist form at the same time. But what about for species where we have no living representative of any kind that can fill that niche, such as the passenger pigeon? That's where biotechnology and other forms step in. Now, this didn't start with biotechnology. The Quagga project is one of the earliest forms of de-extinction project in which they wanted to just recreate the unique coat coloration of an extinct species, that incredible zebra at the top. And it was found that some zebras that live nearby its original environment had fewer stripes. As you can see, the quagga has no stripes on its legs and no stripes kind of on its flank. They diminish. It's got that interesting coloration, and they started just selective breeding. So they've created a new quagga. And if you look up this project, they've been doing this since 1987. They're on their 10th or 11th generation now. They have animals free living in the wild that look extremely similar to the extinct species. Um, it's really, really cool. It was very inspiring to me when I was young. A similar project to try and recreate the auroch, the original type of cow, has been going on by one group or another for decades, and maybe eventually they'll do it. Um, but basically, all living cattle breeds are descended from the original wild auroch, so they all carry its genome in various ways that have been manipulated through selection. And so the idea is to mix those breeds together in a way to undo that selection and get back to the original form. This is kind of interesting because living cattle really are aurochs. They've just been transformed by people once already, so we're just transforming them back. So you have the quagga where it's just a look-alike, then you have the auroch where it's an actual descendant. The new species will be a descendant of the original. Um, for most endangered, I'm sorry, for most extinct species, we don't have living descendants or lookalikes to do that with. Therefore, we're using gene editing technology to get them back. This creates something new. So the new passenger pigeon will not be an original passenger pigeon. We cannot revive that original animal, but we can revive its genes. And those genes can end up bringing back its traits. And this is basically how this works. This is a roundabout way of talking about it. The first engineered animal ever is considered the red canary. And this was just done through breeding. Some German scientists wanted to take yellow canaries and make them red. And they took red siskins from South America and they interbred them. And they had a very functional understanding of genes, even though this was before DNA was found to be the inherited, how DNA worked had yet to be figured out. But they knew that you would combine traits from the two species and that they used to call genes factors. So they called these red factor canaries. And so when the hybrids breed, these little bars here are examples of chromosomes, little cartoons of chromosomes. So that hybrid has one chromosome from the yellow mom, one chromosome from the red dad. When it breeds, those chromosomes will undergo meiosis and recombine. And now we get a next generation hybrid that has chromosomes that are made up of bits of red siskin DNA and yellow canary DNA. So we're getting an animal that is, if you backbreed it, more yellow canary, less red siskin. And if you select for just the right trait, eventually you carry over just those genes that create red color in the background of a yellow canary. And so we've moved the genes from one species into another with design and intent That's why this was considered kind of the first engineered species. There's a fascinating book about it I highly recommend. And this is very similar to what the genome of a new passenger pigeon would look like, only we can't breed this animal. It's dead, so we sequence its DNA and we can introduce a few genes. We're not going to be able to do everything, but we can introduce a few key traits from passenger pigeons into the genome of a band-tailed pigeon. And if we do it under the right selection of those genes, then hopefully the animal we make will have the same behavior and look and ecology of the extinct passenger pigeon. These types of kind of hybrid selected genomes are not rare in nature. They are not just something we create in aviaries or labs. Red wolves of the East Coast have been formed through hybridization of gray wolves and coyotes that have mixed with a third lineage to create them. Every brown bear in North America has polar bear genes, and every non-sub-Saharan African human being, anybody from Europe, Asia, etc., just so happen to have genes from Neanderthals, some of which have been under selection to give us the traits that we have today. So we're a lot more like a de-extinct animal than we might realize. So there's a lot of research we're doing now. We've been working for a decade without much money, and then in 2022, we finally gained a major donor. So we've been able to launch all the science that we've been building up towards over the years. We are sequencing genomes from a bunch of band-tail pigeons and a bunch of passenger pigeons and comparing a lot of variety in those with the newest sequencing technologies that are available. This is a huge improvement on our original 2017 genomics work from UC Santa Cruz. And we're doing this in a particular approach to identify the traits we care about. So passenger pigeons were incredibly unique at the center of this diagram. They were sexually dimorphic, so the male and the female were different color, which is rare in pigeons. Only about 15% of the world's 320 species of pigeons are strikingly different in color between the male and the female. Most of them look exactly the same. It also had a graduated tail, the only North American pigeon to have one. The only other species are the doves, such as Zenata in North America. That's a rare trait. Only about 9% of the world's pigeons have a graduated tail. And then, of course, it was a hypersocial colonial breeder. Only about 4% of the world's pigeons do that. And it was the only true colonial breeder. It bred like a seabird out at islands, right? In these giant flocks all the time. Obligate colonial breeder. The other colonial breeders we have are not kind of true there. And we're going to sequence genomes from a bunch of species that share those traits to try and figure out what is the evolution, what are the pathways on genes that create those traits. And then we're sequencing the genomes of a whole bunch of its living relatives, both ancestral and sister, the living North American pigeons and the cuckoo doves of Southeast Asia to get an idea of what truly made it unique among its relatives. And that's happening right now. We've got genomes of about 50 some genomes of about 18 species total that are going into this analysis at New York, being done by an amazing scientist named Simona Secamandi. Once we have those genomes, we have to be able to create baby birds. So we have work on something called germline transmission. We can't clone a bird, but we can take the stem cells called primordial germ cells from an embryo that become sperm and egg, and we can take those out of an embryo, grow them in a petri dish. We can then implant those into a surrogate mother and a surrogate father. So the surrogate mom carries the germ cells that we've engineered, and the surrogate dad carries the germ cells. They make sperm and eggs that are engineered and breed and produce the baby we want. We're also looking into whether or not we can use induced pluripotent stem cells from feathers and other sources to create germ cells, or use embryonic stem cells to do this. We have 3 or 4 labs working on this around the world. And we are also trying to figure out how we engineer the types of traits we care about. The traits that make a passenger pigeon unique were its behavior, and its tail, its plumage, and its color, as well as how quickly it grew and things like that. We can't engineer pigeons until we can figure out how to grow those cells really well. We're getting close. One of our labs in New York has been growing some pigeon germ cells successfully for a couple months now. That's a huge step forward. But then we have to be able to keep those cells alive long enough to do gene editing at scale. And so we are using the chicken as our model organism to study how to do de-extinction. We will take the living chicken and we are going to change its behavior, plumage, and physiology to make it look like the original red jungle fowl it comes from. And if we can't do that, we can't hope to try and transform a band-tailed pigeon into a passenger pigeon. So we got to be able to take these baby steps forward. This project is in its entering its second year. It's just a week away from doing the largest gene editing experiment that's ever been done in a bird in history. So we're excited about that. That's happening in the petri dish. We're hoping next year it moves to real birds. And it's a cool 5-year program. All this science has to come together. We need a blueprint for the species. We need the cells to work on. We need to know we can do this at scale. It's all currently scheduled to come together sometime in 2029 so we can start engineering cells of the living band-tailed pigeon. And hopefully by around 2032 or so, we've transformed that bird into something that looks like a passenger pigeon and acts like a passenger pigeon and will fool a naturalist into believing we've really genuinely brought it back. And hopefully we'll even fool the trees into believing that. And the true test, if only we could talk to a tree, because there are trees out there in the eastern forest that are a couple hundred years old, some of them 400, 500 years old, that actually once had passenger pigeons in their branches. And hopefully we fool them into thinking that we've truly revived the passenger pigeon. And it's that ecology that we care about the most. So, as I said, no one in the 1800s ever studied that in person. So, we are working to simulate a nesting event. We're not just paying for biotech development, we are studying the ecology of the forestry here. And so, in Wisconsin, probably in the next 4 to 5 weeks, we will be taking a team of volunteers and going out to a patch of small forest, breaking branches, removing seeds and berries, and we will be bringing in truckloads of pigeon guano from South Carolina to Wisconsin and dumping it on the forest. And we are going to be fundamentally looking at how it changes soil chemistry, the microbiome of the soil, how it changes how invertebrates and macro species use that environment. We're even going to be measuring how the carbon cycle changes in the atmosphere of that place. We'll be doing core rings about how the trees respond. This year it will be a small pilot project that starts so that we know how to do all the steps so that next year in 2026 we start an experiment that covers maybe 2 or 3 acres in guano and we'll do observations over about a 5-year or more period so that by the time we are making real passenger pigeons in an aviary to breed, we've got a really strong sense of what they're going to do to the forest and how to restore them because this data works 2 ways. It informs us more about what the pigeons did in the past. It informs us on their impacts they'll have in the future. All that data combined gives us insight into how best to reintroduce them to the ecosystem so that we can take a history of unintended consequences that have taken us from a bioabundant and biodiverse world to one that's quite impoverished today and form a future of intended consequences where we have all of these things back thanks to conservation and biotechnology. And if you think this project is cool and our other 80 projects we've helped and the 50 active ones today we have are cool, I highly recommend going to our website. We are philanthropic and donate to us. We rely on donations from anybody and everybody. And thank you very much. And I'm happy to answer any questions about anything in the program.
Well, thank you,
Oh,
Ben, so much.
yeah. Yeah. Thank you so much. And let me remind everyone tuning in, if you have questions, please throw them in the chat here. Uh wow, this is absolutely a mind expanding concept. Well, I have a whole bunch of questions, and the first one I'd like to ask is what's the critical mass? Like, how many passenger pigeons do you need before you have a flock that's big enough to start making changes to the landscape?
Yeah, we don't know. That's something we're hoping some of this forestry research might actually be able to help us with. So that we're going to dump a certain amount of guano on the habitat. We can equate that amount of guano to a certain number of birds. Um,
Mhm.
And so we're going to see what the effect is at least two different levels of guano. And we'll start kind of trying to simulate from there, hopefully. Because we don't know. We don't know if it's going to
Right.
be just a few tens of thousands of birds that start making an impact at some local scale, or if it needs to be millions or a billion. And that's what long term is kind of exciting about the restoration part of this project is that there's multiple levels of success, right? So for success one is a bird that looks like passenger pigeons hatching in an aviary. Success two is knowing that those birds behave the way we want them to behave. Step three is doing soft releases and that those working. Success four is taking the reins off and saying we're going to let these animals go and do their thing. And then the fifth one is getting them to a self-sustaining population level. And that could be very low. Something like
Mhm.
10,000, 20,000 birds could be self-sustaining. But we don't know then at what level they become ecologically important. Um, we know that we're also trying to maybe figure out what the carrying capacity of the ecosystem is. I'm comforted by a forestry scientist that told me 10 years ago there's plenty of forest for the pigeons. I mean, he knew his stuff, and that's how he said it. But and then, of course, there is the number that's socially tolerable by people. Um, I don't really worry about that one too much, and that's because my kind of dream for the future of this is that through doing this project, it gets human beings to a point of actually learning how to adapt our behavior to live with biodiversity and bioabundance rather than forcing biodiversity and bioabundance to conform to our world. Um, and we have a century or more to do that. It's going to take a long time to get millions, tens of millions to anywhere close to a billion birds, right? So
Mhm.
So I think the long term of this is really the idea that even just thinking about how do we live with a billion birds again in the most population dense part of the nation um, is something that will force us to just become better human beings for all the other conservation goals we care about in general. Really quick, I want to address some of these other questions.
Sure.
Um, are you a 501(c)(3)? Yes, we are. So that we are like the official actual nonprofit, not the ones that get away with some loopholes. Um, can you restore the passenger pigeon without restoring the American chestnut? Yes, we can. So the passenger pigeon was a generalist. It ate pretty much anything. Um, and I didn't include some of the data we had from, 5, 6 years ago, but our first kind of cool discovery in the work was learning that passenger pigeons have been abundant in North America for anywhere between 50,000 years to maybe a million years ago. And that means they were abundant even before human beings got here, but that means they were abundant when species like chestnut and oak were really rare, and conifers were actually dominant. And it means that basically they were shifting their diet whenever the environment changed, and they were noted to have eaten the seeds and berries of 42 different genera of plants. So absolute generalists. They don't really depend on any one thing. Um, Getting their flocks to be big again in today's forests will certainly rely on things like oak, because of their large masting crops. Um, but it's they lived in abundance when there was rare oak too. So we'll get there. What stages come later in assessing the future post-release and how will biodiversity change over time be monitored? Um, Monitored the way we do everything. You guys go out. I assume you guys go out and do your Christmas bird counts, right?
Yep.
Well, hopefully on the East Coast, when they go out in Christmas time, somewhere they see a flock of passenger pigeons going by. Um, what's cool about what I want to do, and what a lot of people are starting to do, is that over the last 20 years, people started tagging data loggers onto species and learning where they go. And those were the kind of things where you had to go and then retrieve them afterwards and record the data. But there are now live feed data devices that you can attach to species, and they're getting smaller. So when we start releasing passenger pigeons, we want to know that they're actually flocking the right way, that they're not scattering, that they're not starting to behave more like band-tail pigeons, but they're actually maintaining the behavior and ecology that we want them for. And so our hope is that every single passenger pigeon eventually released in the wild will have a microchip that I can sit down at a laptop and literally just watch where they're at and where they're moving in live time. Assessing the impacts of the environment will take more things like maybe LiDAR technology, other technology, but also just a lot of people in the field on the ground measuring change, which we hope to be able to sustain as well. Um, can you
Mhm.
show the slide of Saturday,
Oh,
June
that
28th?
yeah,
Yeah, we'll dig into
Yeah,
yeah,
that later. I'll
yeah.
post
For
it
the
in.
person that wrote that, as you see, if you just go to our website, PasadenaAudubon.org,
Yeah,
you can get
I
all the
would show
information
you.
about the.
I would show you, but that's not my slide.
Right. Yeah. Let me ask you a few more just nuts and bolts questions. Hey, when you get ready to release these things, uh, I imagine you need all sorts of permission. How does that work? I mean, who do you? I guess you talk about fish and wildlife. Do you talk to individual states? Do you just open a cage in the middle of the night and hope no one
Yeah.
notices?
Oh, my plan is to become president and then just do whatever I want.
Good.
Um,
Okay. Well, you've got my vote. Yeah.
so the no, it will absolutely have to go through, some type of government permission process. And really we don't really know what that is yet.
Mhm.
Um, no policies were really made for releasing any kind of genetically engineered organism into natural environments. But that's
Right.
something that is coming quickly for a lot of different applications, from microbes and remediation to biocontrols to things like not just passenger pigeons, but like say genetically engineered black-footed ferrets, like what we're working on. So engineered and gene edited wildlife is coming, and probably before we are ready to do some types of releases, there may be some actual clear navigated pathways there. My hunch is that I'm going to have to start making visits to DC and helping make that happen myself. Um, but I think our actual Planet Revive and Restore, because is to develop another nonprofit organization that strictly does passenger pigeon restoration, everything from the socio-political dynamic to actual on the ground. Um, and that is because there's a fundamental shift when you go from creating the bird in an aviary to wanting to breed them up for restoration. And that shift is cost. So a lot of people think de-extinction is really expensive. And the biotech parts of it, yeah, they're expensive. They're nowhere near as expensive as repopulation. Um, and so, we will probably recreate the bird somewhere in the ballpark of 15 to 20 million dollars. Um, Once we're breeding them, it will be easily $100 million a year, every year. Anywhere, it could go drop down to say 10, 15 million a year, depending. But breeding programs for like the California condor or whooping cranes or Hawaiian honey creepers, they cost anywhere in government money from 15 million to $100 million a year. Those species that have been being bred for 40 years in captivity have accumulated anywhere from 1.7 to 4.3 billion dollars in federal spending, which sounds like a lot, but you have to remember those numbers are not even a drop in the bucket when it comes to government spending. Now,
Yeah.
those programs have probably spent twice that because of organizations like the Audubon Society, zoos, and other nonprofits that also then put their own money into these things. Conservation done right requires a lot of people, and that personnel time um, costs money. But that's also why you can look at it in terms of it's going to cost $100 million a year to repopulate passenger pigeons, or you can look at it as in we're creating $100 million of economic stimulus in the jobs and people that do that. Um, so you know, I don't know if that really answers the question so much, but we at Revive
Mhm.
Restore, we try to catalyze projects for other people to take over. We create tools and then conservation groups we hope adopt them, or we were trying to make that facilitation happen. but we don't raise $100 million a year. That's the kind of thing that I think will require Forest Service money, Fish and Wildlife Service money, Department of the Interior money, basically. It might require a much larger nonprofit that's member based. Um, and so our plan is to try and create that nonprofit that is dedicated to this project. It's probably gonna be a lot easier to gain interest in that with hopefully support from millions of people when there's a bird to look at rather than just something that's theoretical. But anyway, we have another question here. Did you say you're
Yes.
using actual passenger pigeon DNA? So, well, um, technically, yes. So we are sequencing passenger pigeon DNA from passenger pigeons. But once it's sequenced, it's just digital data. To make it living again, it has to be synthesized. So the molecules that will go into a band-tail pigeon genome will be the same code as passenger pigeon DNA to transform its genome, but those will be molecules that are brand new, that have been made new from that original digital sequence. Because yeah, once you
And you're
sequence DNA, it's fried, it is done.
right. And your source for this original DNA, I assume, is there are a number of passenger pigeon specimens in
Yeah,
museums around the world.
yeah, and this is something that I probably should have worked into my presentation at some point, but what I think makes the passenger pigeon really the world's most ideal de-extinction candidate is one, yes, it theoretically may have a very important role in the environment, possibly the most important role in the eastern U.S. forest environment of any species. Um, but to be able to restore a species, you need to know a lot about it. Um, and we fundamentally just know more about passenger pigeons than any other extinct species in the world. And that's because one, they were a industrial harvested species. Companies hired people to go out and hunt them in mass. And to be a good hunter, you need to know your prey. And these people that were going out and trapping, they took notes. We know over a dozen roosting and nesting sites. We know their approximate size and shape on the landscape. We have, I think, 2,000 observation data points across the eastern US from over 500 years. Lots of personal accounts. They were bred in captivity by one person. And so we have an aviary timeline of how long it took to incubate the eggs and build the nest and courtship. We even have notes on how the males danced for females. Um, to be able to verify the records we get from the hunters and trappers that say, oh, it took several weeks to build the nest, then how long the egg went. And they cared about that because they wanted to know when were there fresh babies to go and harvest, when the males left the nests in the morning to go feed and then come back to the females. We have so much information that was never collected for scientific purposes, but was collected in almost a very scientific way. And so we can reanalyze that. And then when it comes to that genome, there are 1,500 preserved passenger pigeon skins in museums around the world and in private collections. Um, that's 1,000 more skins than any other recently extinct species on the planet. Um, so yeah, the number two is at 500. So we've got more sources of DNA. We've even pulled DNA from bones from 4,000 years ago to help kind of complete our population analyses. So yes, we've got a lot of stuff to look at, and it's that amount of information that will allow us to succeed. Um, let's see. Have you received any pushback from any organizations with regard to any ethical ambiguities? And if so, what have their concerns been? Environmentally speaking, what are the possible risks, drawbacks, et cetera, et cetera, et cetera, blah, blah,
Okay.
blah.
Actually, I wanted to ask you about one of the arguments, and I'm sure you're very familiar with it. One of the arguments about de-extinction is that it will make us care less about conservation. That if there's a sense that we can bring back anything that we destroy, we won't take care of species as well. When someone hits you with that, what do you say?
Well, I had a different answer
Oh, okay.
seven months ago.
Right.
Um, I don't know how much you guys have been following the news, but this moral hazard of de-extinction will make us care less about conservation in the present has materialized in front of our very eyes in the last month. Um, my answer to it technically has not ever changed and still hasn't changed. It's just become more alarming. We used to kind of not really laugh, but we thought it was somewhat laughable because the reality is this is just true of conservation. You're never going to convince someone who doesn't care about nature to ever give a shit about it. It's you're either born with a mindset that you care about this, or you're apathetic and you're just trying to get by each day because of the whatever's going on in your life, which sadly in this nation is usually not enough money to go around, despite the fact that we have so much money. Or you're one of the people that actively loves destroying nature because it makes you rich. And we might be able to pull some of those apathetic people to our cause and say, you know what? Experience nature once and fall in love with it. And it doesn't have to be a wilderness, right? It can be the birds in your backyard. It can be actually bothering to lift up a rock and see the incredible snails and isopods underneath of it. But those people who get rich off of destroying nature, they're never going to care.
Mhm.
And for a long time, the majority of people in the United States have cared about sustaining nature, at least in so much as being able to say on a poll, like, yes, I support the Endangered Species Act. But now we have an entire administration filled with people that basically don't care about nature. And 3 weeks ago, I think it was 3 weeks ago, the Secretary of the Interior said one of the most appalling things anyone in government could ever say in our field, and that is thanks to the recent announcement that the direwolf was recreated by a for-profit company, which that's a lot to unpack there. You can look at any of the articles online to see what a lot of scientists have said, and most scientists have been saying the reality that this was kind of a overcrazed media hype of a thing. But the Secretary of the Interior really celebrated it. And initially, I wanted to be happy about that because I was like, okay, here we have a government that might be willing to want to integrate biotechnology, which is really needed in conservation for species today. Not every species need it. The number one thing species need right now is habitat, and that's basically it. We need lots of habitat. We need high quality habitat. We need to remove harmful invasive species and diseases, get rid of pollutants. We need to restore corridors. It all comes down to habitat, really. But in many cases, biotechnology will play a role in making that habitat livable, and it will play a role in saving the species we want to save. So that was cool. It was like, okay, here we go. But then he gathered up everybody in the Department of the Interior and he said, we no longer need to anguish losing a species. Just call up Colossal and we'll bring them back. And
Yeah.
that's incredibly frightening. Um, Revive and Restore has issued statements to basically reject that philosophy and refute it. Because if you had asked me 7 months ago, I would have said the same thing. I would have said people are never going to care and people will. And it's something that's never materialized before. When people started breeding animals in captivity to restore them, when the California condor is a great example, there was an entire non-organization that came about called Extinction with Dignity because they felt condors should die in nature because they didn't think there was enough evidence that humans were causing their decline. And they felt that it was so morally reprehensible to bring these birds into a zoo and let them live their last days in a zoo rather than in the wild. And they had the same argument. They said, once you start doing this, it's going to be a zoo animal forever, and no one's ever going to want to conserve species. They'll just take them out of the wild, put them in a zoo, they'll pave their habitat and go. And never before in 50 years had that actually been a real risk. It never played out. And yet here we have a government that's using biotechnology and innovation
Yeah.
as a greenwash to undermine the Endangered Species Act. So They were going to do that no matter what, but it is extremely concerning and frightening to see the thing I've been advocating for 10 years as a good thing be used to fundamentally gut our biodiversity
Yeah.
policies here in the United
Yeah.
States.
Yeah. Well, oh yeah, a lot to think about there. We got another question in the a quick question perhaps in the chat. Have there been any issues with CRISPR engineered organisms? I don't know quite what they mean specifically.
Yeah, well,
Yeah.
there's two classes of has there any been anything issues, right? So there's the issues of to the organism itself, have the gene changes made problems there? And then there's, of course, are there then problems with whatever that animal's interacting with?
Right.
Um, at the moment, there is only one gene edited organism that's in a natural environment. That's a tree in Georgia. Um, I don't think anything bad's happening so far. Um, with genetically engineered organisms, most of the ones that have been made are to study how genes work. Um, and so technically, some of them have had what you would call issues, but that was kind of the point. They were trying to figure out, is this gene critical for a particular pathway in the organism? Um, so, one of the most common things people do to even test if gene editing is working is turn an animal albino. Now, an animal that's albino can live just fine in captivity, but you might not want to release it to a wild environment because albinism is not so great. Um, but yeah, so it depends on what the research has been doing. But in general, if you're gene editing genes that you already understand to create, say, gain of function or other traits that are healthy, you can produce healthy offspring that way, and they live just fine. Gene editing itself is it all depends on the gene that you're messing with and what kind of
Mhm.
change you make. Um, so yeah, I have not heard of any real significant problems with gene edited organisms so far. But
Okay.
once again, yeah,
Uh,
it's
Sarah
okay.
points out we skipped over the second half of a question she posted, which was
Oh,
environmentally
yeah, yeah,
speaking,
ecological
what are the
risks.
possible risks or drawbacks? Yeah.
Well, for passenger pigeons specifically, the risks we worry about are going to be when they become abundant, is to whether or not that abundance can be managed in a way for benefit. And so theoretically, it should, right? I mean, if you imagine, I showed you that diagram, like it looks beautiful on paper, right? Like, oh, there's all these species declining today that need these disturbances. And so once you start those disturbances, it should work great. But the problem is because our populations are fragmented, our viable habitats are fragmented, and some of those risk species are now really rare. The thing that's supposed to come and save them could be the thing that ends up actually killing them. Um, because imagine that you now have a plant that really needs those first successional forests and habitats to thrive, and it's really rare now. And then you have a flock of passenger pigeons come in and dump a foot of guano on top of the last population and kill it. That should have been the thing that ended up creating habitat for it that's ideal to help it thrive. But because of the circumstances of how the environment has changed, we need to plan for mitigation. And that's why monitoring the movements of the species as well as their population size becomes so important. If this population size goes up slowly, theoretically, which it will, there's really no way to create a billion birds fast. Um, not even if you threw $1 trillion at it, you could not do this fast.
Mhm.
Um, the you know, it relies on the fact of maybe there are certain regions of the forest where we don't want them to create disturbances until we've been able to secure the populations of the species we want to save there first, right? It's it really comes down to do we need to move a species out of a certain area first, bring it into a zoo, something like that. And that may never materialize as a real risk, but it's something that you have to be ready for. Because, yeah, after 100 years, this species should be creating habitats and doing something really great. But all those species alive today that once lived with passenger pigeons for tens of thousands of years and who should benefit are living in a state today that is really, really, really different dynamically. So yeah, it comes down to planning. So there are risks. I don't think any of those risks are unavoidable.
Mhm.
Um, yeah, particularly if the species were to start populating too fast. This is really, really crass to kind of say, but like we already know how to kill all of them.
Right. Yeah, good point. Yeah. Yeah.
I mean, gosh, we got way better guns today than they had 150 years ago, guys.
Right. Do we have actually one thing that sticks in my or one image I have is a flock of 500,000 passenger pigeons uh defending and descending on some suburban area like Pasadena. And do we know from historical records? I mean, there was obviously way less suburbs, 150 years ago, but there were towns and stuff.
Yeah.
Would they descend on towns too, or they pretty much stayed out into full on forest?
So this is where the historic accounts are interesting, but once again, not necessarily very informative, right? Because we're not reaching into the past and resurrecting a passenger pigeon from 1860. We're engineering a band-tail pigeon. So if we want to know a
Mhm.
lot about how this species might interact with human environments, we have to look at how band-tail pigeons interact. And I think when we got on the call, you said you had a couple in your backyard, right?
Yeah, yeah,
So
yeah. They're very common in the suburbs around here. Yeah.
yeah, yeah. But what we do know about band-tail pigeons, though, is that they also do like to breed and stick to more intact forest area.
Hmm. Yeah.
So they're a bit of a suburban bird and
Hmm.
still a wild bird. So it's possible our new passenger pigeons are going to get that from band-tail pigeons where they might come into suburbs. And this is where the growing pain happens, right? Is that technically, if we repopulate them fast enough, the flocks will be too big to sustain themselves on suburban environments.
Uh-huh. Yeah. Mhm. Right.
But there will be a pinch point, right? Like, yeah, it's possible they're going to go into a suburban park and destroy it with their guano, and that
Right.
would be great. Historic passenger pigeons did not do that.
If we could breed them to selectively attack golf courses, that would be
Cool.
fabulous. Yeah.
Yeah, so that's the thing is like historic records of passenger pigeons show that they did not like human-made structures. They
Mm,
avoided.
okay. Okay.
The only real records of them descending upon human-made structures was during fog or storms. They flew overhead of towns, like they absolutely flew over, but they were not
Sure.
landing next to them. They were going
Yeah.
out to big patches of forest. But once again, that was probably because if you're trying to land a billion birds, you can't do that in Central Park, New York. Um,
Hmm.
and even if you can, they still have to feed. And the closest food outside Central Park, New York, is 30 miles away. Right? So I think it gets back to the point of we need a big enough flock that it really can only sustain in large wilderness areas. Now that becomes an issue for all the people that live out more rurally, but hopefully in 100 years, none of those people exist. So I say it as I am a person who lives in a forest in North Carolina.
Yeah, yeah, yeah. You're going to be covered in guano here if everything goes your way.
I hope
Uh,
so.
yeah.
Um, but there will also be, I think, the thing is, I'm from North Dakota originally, and I went to school in Montana. And this isn't the
Hmm.
best equation here, but if you think of Kalispell, Montana, that city,
Sure.
beautiful to visit, definitely
Yeah.
go. Or Anchorage, Alaska, right? Like these are modern urban areas, and they live with mountain lions, wolves, grizzly bears, moose, elk. They live with big dangerous and property damaging species. And they walk right through their yards and nobody bats an
Mhm.
eye because they've adapted to living in the midst of nature. Now, that's not a billion passenger pigeons coming and pooping, but humans can coexist with the wildness of nature. And we have so much technology to be able to potentially deter birds from landing in our cities and change the way we build our homes, our parks, and other things to be more compatible. And that's once again the reason I kind of really love the idea of this project over the course of a century is that it will push us to think about if we really want to achieve conservation goals, and this is true, Like, if we keep going the way we're going, saying, well, we're going to save this species here in this wilderness and this one here in this pocket, we're not going to succeed. It's every species is just going to be on a lifeline that depends on human beings forever because we're not giving them enough space and coexistence to thrive with us. And what better
Mm.
emblem of pushing us to coexist with nature than having a billion birds fly overhead again? If we can do that, then we should be able to accomplish any conservation goal we set our minds to.
Okay, I'm sold. I've got a quick question here before we let you know. I didn't want to let you go before finding out what was the name of that book that you'd mentioned about the red
Ah,
siskin. Yeah.
where is it here? Oh, now it's not on the shelf.
Hmm.
Um, Gosh, it's probably in a pile right over there.
Okay, well, I can.
Anyway, so it's a. I'll just Google it really quick. It's actually
Yeah.
called. Let's see here. There it is. It's called the Red Canary.
Okay, I'm throwing this in the chat for anyone that cares. Okay.
Let's look at this here. Let's get it on Amazon. Here we go. So here's a link. It's by Tim Burkhead.
Oh,
Fascinating read. Um, Really fascinating. And I like using that as
okay.
an example because
Okay.
the red canary is not only the first engineered organism, but it's also an organism that gives us the insight of the fact that a genetic code only gets you so far.
Mhm.
So the man who made the red canary died thinking he had failed because he was a firm believer that genes create our traits 100%. And as we know today, genes are a blueprint for traits. They are created by the environment, everything from the cellular environment, the womb, to diet, to the exterior environment shape the traits of an organism. Um, and the thing is he could never get his red canaries to be red. They would only be orange.
Hmm.
Um, and this is the thing. So if you have a regular yellow canary and you feed it a particular diet, you can get it to be orangish and
Yep.
almost red, but you can't get it to be pure red. If you have a canary that has a gene for being red, you can get it to be orangish, but if you don't feed it the right diet, it will never be red. It's a combination of the two. So the only way you get a pure red canary is you have to have a gene that takes yellow carotenoids that come from the diet and transforms them into red pigment. If you don't have the gene, it doesn't matter how much red pigment or whatever they eat, they'll only deposit so much of it, they won't be pure red. So you need a combination of the gene and the diet.
Mhm.
And that's exactly what I think is going to happen for these behavioral traits is that we can give a whole bunch of genes from passenger pigeons into bandtail pigeons, but if we don't raise our first babies with the sights and sounds of a flock of millions of birds, we probably won't elicit the expression of those traits the right way. So we're not even going to do the science behind that. We're not going to have a group of birds where we don't do it and a group of birds where we do. We just want to hedge our bets and just be like,
Uh-huh.
there is an environmental factor that matters here. We're going to simulate that. And that's how actually colonial breeding seabirds have been reintroduced to islands and things. So
Sure. Yeah. Yeah. The puffins in New England are a great example of that. Yeah. Yeah. Do we actually have recordings of the sound of passenger pigeons?
no recordings. There is a book chapter that has kind of what people, wrote down what it sounds like, but
Hmm.
we probably will never know if we recreate the sound.
All right. I see. Well, I think we're out of questions here in the chat, and I think this we've been I've been thrilled to have you here. I've wanted to learn more about this for a long time, and I'm very excited. I usually just be excited about it as it'd be a new species on my my life list, but but the fact that it could also revitalize the force of the East. I'm from the East myself. Uh, Sign me up. I'm totally on board with bringing back the passenger pigeon or
Well,
recreating the passenger pigeon.
I will share one more link in the chat for everyone. This is something
Okay.
I was personally really proud of. So it's not a whole lot about the science, but it's a really cool show. It's a 4 episode series that came out last March, or March of 2025. On YouTube, it's backwards. So I'll actually share my screen again really fast just to show you. If you click
Okay.
on the link, this is what you'll see. This one down here that says number 4, that's episode 1. So it's
Okay.
in backwards order for some reason, but it's 4 episodes all about the people around the world that are involved in basically biotech and conservation, and has a lot about the people working on northern white rhinos, and it follows my work and my story for all 4 episodes.
Oh,
Um,
great.
and it is actually much more about the people, um, and their stories and what drives them. So it really humanizes all of this. Um, because nobody does any of this work without personal reasons. It's science is a beautiful philosophy, but it's done by people. Um,
Yeah.
and yeah, so it's a really nice glimpse into my life and everybody else doing that work, and it's also it's a beautifully shot series. So
Oh,
highly
well,
recommend.
fantastic. Yeah, thank you. And I'd like to mention to everyone here that uh, Ben, you've kindly given your permission for us to record this talk, and it'll be up on the Pasadena Audubon YouTube channel in just the next few days. So you can, by all means, watch it again, share it with your friends, and so on. And with that, Ben, we'd like to thank you so much for taking the time tonight. And Jared, back to you, man.
Yeah. Well, yeah, thanks again, Ben. Thanks, Chris, for putting this on. And thank you all. We have one more monthly meeting before we take our summer break in July and August. So we'll see you in June. I forget the exact date of it, but it's the third Wednesday of the month. Um,
Yeah, June 18th.
June 18th. And then we'll see you again June 28th, just 10 days later at the June banquet. So thanks, everybody. Thank you, Chris. Thank you, Ben. That was truly fascinating and excellent work. All right. Have a good night, everybody.
Good night.